All right, good afternoon, everyone, and welcome to the Genetics Colloquium. It's my pleasure today to introduce Dr. Hongyan Zhu from the University of Kentucky. Hongyan did his PhD with Dr. Doug Cook when Doug was at Texas A&M University. And then his lab moved to the University of California, Davis, and Hongyan followed Doug over there, and Hongyan continued on to a postdoc with Doug Cook for a few years. And in 2004, he obtained a faculty position at the University of Kentucky. He moved through the ranks, and now he's full professor, published plenty of papers on, in particular, rhizobium legume association, that great symbiosis that leads to root nodules and nitrogen fixation. And Hongyan published lots of genetic studies. That's why I think he's such a great speaker for the Genetics Colloquium, in particular on the plant side, but more recently also genetics on the bacterial side. And initially, I think he pioneered also the work of trying to compare the mechanisms between legumes and cereals and rice in particular, publishing lots of papers on showing that the mechanisms are actually quite conserved between that rhizobium legume symbiosis in legumes and what we call mycorrhizal association in rice. And that work was really a pioneer, I think, to pave the way for engineering root nodule symbiosis outside of the legumes, because he really showed that these mechanisms are very similar between legumes and cereals. Today, I think he's going to talk more about specificity in rhizobium legume association. He did lots of, again, great genetic work using natural diversity, and particularly on the plant side, and doing great genetics. So with that, join me to welcome Hongyan to Madison.
Thanks for Jean-Michel for the nice introduction, and thank you for joining this seminar. Currently, I have really enjoyed to talk with faculty and students. So my lab's research focuses on genetics of symbiosis specificity in the legume-rhizobia interactions. So first, give you a little bit of background. We all know that nitrogen is essential for life, but it's also a limiting nutrient for plant growth. So even though nitrogen is very abundant in the air, but not directly available for plant, it has to be converted into ammonia so that plant can use. So modern agriculture has been heavily relied on use of chemical fertilizers. So production of chemical fertilizers consume a lot of energy. So it represents a single most energy costing aspect of agriculture. A big problem is that, when you apply nitrogen fertilizers, only 40% of the fertilizers are actually taken up by the plant. The remaining is run off to the waterways and also generates a lot of greenhouse gas. So a significant example is the dead zone in the Gulf of Mexico. It's affect the marine life in that area. So in nature ecosystem, the plant of the legume family can form a symbiosis with soil nitrogen fixing bacteria. The resulting of this symbiosis is to form nodules on the plant root. Within the nodules, the bacteria use the nitrogenase to convert nitrogen into ammonia. So neither the plant nor the bacteria can do it alone. So that we call symbiosis. So giving an example. So this reaction, the nitrogenase is very sensitive to oxygen. High concentration of oxygen will kill the enzyme. In another aspect, this reaction consumes a lot of energy, and bacteria require the oxygen to generate ATP. So this is a conflict. So this conflict is resolved in the root nodules. You see the root nodule, the color is pink, all right, because of the presence of abundant leg hemoglobin. So leg hemoglobin protein is similar to human hemoglobin or myoglobin. It's an oxygen transporter. So this leg hemoglobin binding oxygen and protect the enzyme, and at the same time to control provide enough oxygen to generate ATP. So first, I want to give overview how symbiosis is developed. It starts with a signal communication between the plant and the bacteria, and the nitrogen limiting conditions produce and secrete flavonoids into the rhizosphere, and these flavonoids attract the bacteria to the root, and also induce the expression of gene required for biosynthesis of a bacterial signal called nodulation factors. When plant can recognize these nodulation factors, something good will happen. So the one hallmark response of this interaction is forming the root hair curling. Actually. So a single bacteria is enough to induce the root hair curling. And within the root hairs, the bacteria is trapped within the curl, and from there, the invagination of root hair membrane leads to form a tubular-like structure called infection thread. Okay. At the same time, node factor perception also induces the cortical cell division to form nodule primordium. So when the infection thread reach the nodule cells, they release the bacteria into the cytoplasm of the cells through a process called endosymbiosis. Within the plant cell, each individual bacteria is enclosed within a plant-derived membrane, okay, to form an organelle-like structure within the called symbiosome. So within the symbiosome, the bacteria further differentiate into bacteroid. So the form that can fix nitrogen. Even though we call symbiosis, actually plant control this process. Okay, so the bacteria actually function like a slave, eat whatever the plant provides them and do hard work for nitrogen fixation. In Medicago, the bacteria finally die. We call terminal differentiation. So not all legumes are nodulated by all rhizobia bacteria. So there's a high level of specificity. In our lab, we studied two types of specificity. One we call nodulation specificity. Another is nitrogen fixation specificity. So nodule specificity happens at early stage of the interaction. So each bacteria species or strain can only infect and nodulate a limited set of host plant. In contrast, nitrogen fixation specificity occurs after nodule formation and bacterial infection, so that nitrogen fixation efficiency differs significantly between different plant rhizobia interactions. So it's well known that nodulation specificity occurs at species level. Okay, different legumes are nodulated by different bacteria species. So this level of specificity is determined by host-specific recognition of rhizobia node factors. So this is a typical node factor structure. The cating backbone usually contains 4 to 5 sugar units. So the arrow indicates the chemical group that usually are very variable, differ between different bacteria. So for example, if here is a sulfate group, is preferred by plants like Medicago truncatula and alfalfa. So if here is acetyl group, it's preferred by P. So nodulation specificity also happens between different plant genotypes within the same species. Here I show you the two soybean lines, L6 and L82. The same two soybean lines show contrasting nodulation phenotype when you inoculate a different rhizobia strain. So in soybean, resistance to nodulation is usually conferred by dominant genes. So for example, resistance to nodulation by a strain called USDA-122 is conferred by resistant gene called RJ2. And resistance to nodulation by another strain, USDA-121, is conferred by a gene called RJ4, dominant gene. So our lab have cloned both genes. It turned out that both RJ2 and RJ4 encode defense-related proteins. So RJ2 encodes a typical disease resistance NBLRR protein. So when we transfer the dominant gene into the recessive genetic background, this gene can completely block nodulation. It's a blue root. We use expressing gas as a transporter gene. So similarly, RJ4 encodes a somatin-like defense-related protein. If plant carry this gene, cannot be nodulated by USDA 61. When this gene is knocked out by the CRISPR-Cas9, the mutant root, which is the blue root on the right, gains the ability to nodulate with this strain. So I want to point out that in both cases, resistance to nodulation is dependent on bacteria type 3 secretion system. So pathogenic bacteria use type 3 secretion system to deliver virulence factor into the plant cell to cause disease. If this virulence effector is recognized by plant R gene, it will cause so-called gene resistance. Okay, the rhizobia bacteria also use type 3 secretion system to deliver effectors to facilitate their infection of the legume host. And sometimes type 3 secretion effector can induce the nodule formation without node factor perception. Okay, these two examples tells us that effector triggered immunity play an important role in regulating host range specificity. Okay, the question you ask, why legume plant involve R gene to restrict nodulation by beneficial bacteria? So I just talk with students, nature is full of conflicts. So the first, when maybe plant just cannot distinguish between friends from foes due to a lack of adaptation. Consider a situation, both pathogen and the symbiotic bacteria secrete same effector to facilitate infection. Sometimes plant cannot tell the difference. Secondly, plant may face a dilemma between defense and symbiosis. Consider a situation, the same gene block nodulation, which is bad, but at the same time, it might confer resistance to certain pathogens. So in this case, balancing selection will keep this gene in nature populations. Alternatively, plant may use R gene to selectively exclude low efficient bacteria, but nodulate with high efficient nitrogen fixing bacteria. So this is the case in the example I just mentioned, the soybean plant that carry the dominant RGF4 gene are actually considered favorable because RGF4 confer resistance to many bacteria strains that are highly competitive for nodulation, but with very low nitrogen fixation. So if plant carry this gene, they are more responsive to commercial inoculant. Okay. So next, I want to talk about nodulation specificity in medicago. So this is different from the soybean I just talked about. In this case, the bacterial partner of medicago, such as Maliloti and Symbiosis medicae, they don't have gene coding for type 3 secretion system. Okay, and furthermore, so even though Maliloti and medicae both can efficiently nodulate medicago truncatura and alfalfa, but some medicago species show preference toward the, different preference toward these two bacterial species. So we identify a medicago locus we name NS1. So this locus restricts nodulation by the bacteria Maliloti strain RM41. Okay, very simple genetics. I'm sorry. I should have okay. So this is not plus plant. This column is not minus plant. So F2 population show perfect 3-to-1 segregation. 3/4 of plants are not minus, and 1/4 plants are not plus. So this locus function to restrict infection by the bacteria strain. So in compatible interaction, there are nodules on the plant root, and you can readily observe infection thread, which is a red color or blue color, so which deliver the bacteria into the nodule cells. In contrast, in the incompatible interaction, we did observe root hair curling as well as nodule primordium formation, but this nodule primordium are not infected because the infection thread cannot be formed. So the nodule primordium contains no bacteria, and because a lack of infection, the nodule primordium cannot develop into big nodules. So this locus encodes two receptor-like kinase. Okay, this is a domain structure of the receptor-like kinase. They have N-terminal signal peptide. They have mullectin like domain, and 3 leucine-rich repeat, 1 single pass transmembrane domain, and an intracellular kinase domain. Okay, mullectin domain is originally defined by carbohydrate binding, but recent research shows that this domain can bind various kind of ligands. So this is organization of the 2 genes in 3 mapping parent. I forgot to mention that we use 2 F2 population to map the gene. Okay, involving 3 parent, 1 not minus parent, and 2 not plus parent. If you compare these 2 parent, I said two copy gene, we call NS1A, NS1B. If you compare these 2 parent, no difference for the first copy, and the second copy is missing in the not plus parent at 20. So from this comparison, you would infer that the NS1B is responsible for the phenotypic difference between these 2 parents. However, if you compare these 2 parents, no difference for the second copy, but they are polymorphic, they differ in the first copy. So from this comparison, it points to the first gene is responsible for the phenotypic difference between NOD- and DZA045. So we go ahead to do the complementation test. So we transfer the NOD- allele, the gene from NOD- parent into DZA045. You can see the wild-type root is a white root, is NOD+, and the transgenic root is a blue root, is NOD-. So it makes sense because there are differences between the two alleles. So when you transfer the same gene into another parent, both transgenic root and the wild-type root produce nodules. Okay, then we test the second copy. So we transfer the NS1B to the A20. So you see NOD+ on the wild-type root, and no nodules are formed on the transgenic root. And when we transfer the gene into another parent, in this case, cannot complement. It also makes sense because no difference between the two alleles. So the conclusion is that both genes are required for resistance to nodulation in the NOD- parent. Okay, missing or mutation of any of these two genes, the phenotype is NOD+. Okay, so consistent with this, we carry out CRISPR-Cas9 to knock out both genes. So knock out NS1A, you convert NOD- to NOD+, and you knock out NS1B, you also convert NOD- phenotype to NOD+. Okay, so of course we sequence this nodulated root to confirm, this gene got mutated, and because CRISPR-Cas9 is working in a continue working, you sometimes you see multiple mutant alleles. Okay, so the expression of NS1A and NS1B is dependent on host perception of NOD factors. So this gene expression of this gene is root specific and only expressed when you inoculate with either compatible or incompatible bacteria. So in incompatible interaction, the expression only restrict to a small number of we call loci where the bacteria colonized. We call susceptible zone. Okay, in a compatible interaction, they only expressed in the nodules. So this suggests that the expression of this gene is dependent on host perception of bacteria nodulation factors. So consistent with, if we knock out the bacteria gene required for production of nod factors, they cannot induce the expression of the genes. Similarly, if you inoculate the plant with a mutation that are defective in NOD factor signaling, the plant cannot induce the expression of the genes. So the question is that what bacterial signal is perceived by the receptors? So consider that these two receptor kinases, they contain more lectin-like carbohydrate binding domain. We wondered, we hypothesized that this receptor may recognize bacteria surface polysaccharide. And this polysaccharide include exopolysaccharide, capsular polysaccharide, and deeper polysaccharide, EPS, KPS, and LPS. So we generate a number of mutants that are defective in production of this polysaccharide. So the KPS and LPS mutant show the same phenotype as the wild type, which indicates that KPS and LPS are neither required for nodulation nor bacteria determinants of nodulation specificity. When we knock out the EPS production, all the interactions are not minus. Okay, so this indicates that EPS is required for nodulation in metacargo. But whether EPS is the function to, if EPS is involved in nodulation specificity is unknown. So there's a scenario that, okay, EPS is required for nodulation in compatible interaction, but the same EPS may trigger defense response in the presence of the NS1 locus. So we have no idea whether EPS is involved in determining the specificity. So we next conduct a transposon-mediated random mutagenesis of the bacteria genome. So this leads us to identify a bacteria gene which is required for triggering NS1-mediated resistance to nodulation. So this gene encodes a protein that contains so-called parallel beta helix repeat, 5 we call PBH1 repeat, and PBH1 repeat are often present in polysaccharide laces. Okay, so if bacteria carry this gene we call RNS1, it causes incompatible interaction in the presence of the NS1 locus. If we delete this bacteria gene, the incompatible interaction become compatible. So this gene is located in a string-specific gene cluster. So this lists the gene in that cluster. So I just want to give you a summary. Okay. So RNS1 is located in a 55 kb gene cluster that is conserved in about 50% of the myeloid string. Okay. So most genes in this cluster are predicted to be involved in polysaccharide biosynthesis. And this gene is not present in the closely related species, legume-rhizobia medicae. And the presence or absence of this gene is correlated to nodulation compatibility with the plant that carry the NS1 locus. Okay. So when we express this bacteria gene in the compatible string that lack this gene, it converts compatible interaction into incompatible. It's sufficient to convert the compatible interaction into incompatible. So RNS1 is a type 1 secreted cyst protein. Okay, but we carry out protein-protein interaction. We didn't detect direct interaction between the NS1 receptors and the RNS1 bacterial protein. So the type 3 secretion system consists of 3 proteins: one is ABC transporter, a membrane of fusion protein, and an out-membrane protein called TOC. When we knock out TOC, the RNS1 protein cannot secrete to the bacteria culture. So we still don't know. It's still possible. We still don't know the ligand of the NS1 receptor. It's possible that the RNS1 function to process EPS or other kind of polysaccharide and to produce an unknown, maybe oligosaccharide, which is perceived by the NS1 locus to block infection. Alternatively, this protein might also function to disrupt the plant cell wall to generate so-called damage associated molecular pattern. It also can trigger defense response to block bacteria infection. So we just published in PNAS, the title basically summarizes this finding. So the NS1 locus encode 2 receptor-like kinases that confer broad spectrum resistance to nodulation by symbiosis meliloti that carry the RNS1 gene, bacteria gene. So actually, we also identify another locus. I just want to briefly mention it. We call NS2 locus, which can also discriminate between symbiosis meliloti and symbiosis medicae. In contrast to the NS1 locus, this NS2 locus will block nodulation by symbiosis medicae strains. Okay, so these locus map to a large gene cluster contain a large number of molectin-like receptor kinases. Okay, the evolution of this gene cluster is very dynamic. You know, there are significant variation in terms of copy number as well as sequence diversity. So the DZA220H, which contain the NS2 locus, you know, was classified as a medicago. But recent research analysis suggests that it belonged to a different medicago species called medicago, I don't know how I pronounce right, litoralis. And this is the species that prefer meliloti, but not medicae. So this shows you when we knock out this receptor kinase, you know, the mutant root can form nodules by the same bacteria strain. So we use CRISPR-Cas9 to sequence the root. We can see when compared with the wild type sequence, there's a 1 BP deletion in the exon. So cause framework shift, knock out the gene. So we also identify a bacteria gene we named RNS2. Okay, so this gene encodes a glycine repeat, octapeptide repeat protein. So if you compare the 2 protein isoforms from meliloti and medicae, you know, the medicae contain 18 tandem repeat of this, you know, 18 tandem repeat of this octapeptide repeat. But all the meliloti strings, these repeat are interspersed by a number of, you see, SGS, GGG, SGS, GGG. Okay, so this causes the structure change of the protein. This is predicted by AlphaFold. It's very popular. I don't know what that means, so I just know there's a difference between the two. Okay, so we are working to test the interaction between NS2 and RNS2 right now. So you can see the same as RNS1, when we knock out the gene, it can convert incompatible interaction to compatible interaction. And also, if we transfer this gene into a compatible bacteria strain, can convert compatible interaction into incompatible. Okay, so I want to spend the rest of my talk on nitrogen fixation specificity. So I want to reiterate that this nitrogen fixation specificity referred to that the same bacteria can form fixed plus nodules or fixed minus nodules depending on the host genotype. And the nitrogen fixation efficiency can differ significantly between different plant bacteria pairs. So this is an example in Medicago. The same bacteria strain inoculate two different Medicago genotypes. One form fixed plus nodules and another form fixed minus nodules. Okay, similar situation also happened in the soybean. Okay, same strain can induce the formation of fixed plus nodules and fixed minus nodules depend on host plant genotype. So what caused the fixed minus phenotype in Medicago? So both fixed plus nodules and fixed minus nodules are infected by the bacteria. And the infected bacteria also successfully differentiate into bacterioids. However, in the fixed minus nodules, the differentiated bacteria seem to have a hard time surviving. Okay, you see many cells are empty because they are quickly killed or degraded. Okay, so this is a bacteria stand with PI. Fixed plus bacteria isolate from fixed plus nodules and fixed minus nodules. You can see the elongated bacteria presents in different time points, 1 week, 2 weeks, 3 weeks. In the fixed minus nodules, you can see abundant elongated differentiated bacterioid at early stage, but they are gradually disappeared at later stage. Okay, so consistent with, we also carry out promoter gas assay for the nitrogen fixation gene called NIFH. You can see in the fixed minus nodules, the gene was expressed at the early stage, but the expression abolished at the later stage. So the conclusion from this is that the fixed minus phenotype was not caused by a lack of infection or a lack of differentiation. It's caused by bacterioid lysis after differentiation. So we carry out genetic analysis of this specificity in Medicago truncatura using F2 and a recombinant inbreed line population. Okay, in the F2 population, you can easily tell which plant is fixed minus and which plant is fixed plus. However, the segregation ratio cannot be explained by a single gene model. So actually, the genetics is not simple and very complicated, involve multiple linked loci with complex gene by gene we call epistatic and allelic interactions. Okay, so in this case, F2 and the real population are not feasible for finally map the gene. Okay, so nevertheless, we use whole genome scan of the real population. We identify a locus we call NFS1, which is strongly associated with the phenotype, but not co-segregating with the phenotype. In other words, we know there's a gene here that regulates the specificity, but we cannot precisely determine the location of the gene because of the interference of other genes. Okay. Fortunately, in the real population, we identified a line we call a residual heterozygous line. at the NFS1 locus. So this line is heterozygous at the NS1 locus in a nearly homozygous genetic background because it's a recombinant inbred line. Okay, the progeny of this line show a monogenic segregation. Okay, so this is good. It's easy to do genetic mapping and clone gene. So this is a progeny of the we call RHL line. So this is a fixed plus parent, this is a fixed minus plant parent, and this is a near isogenic progeny of the residual heterozygous line. Okay, listen carefully. If we just classify the plant based on whether they can fix nitrogen, the fixed plus to fixed minus show 3-to-1 segregation. Okay, so we name the allele from the fixed plus parent as NS1 plus, the allele from the fixed minus parent as NS1 minus. So from now on, I will simply call plus allele or minus allele. Okay, so if you look at the plant carefully, I just said if you classify the plant based on whether they can fix nitrogen, it's 3-to-1 segregation. Okay, if you look at this pot which contains 4 plants, okay, this plant is dark green, very healthy, and this plant cannot fix nitrogen and it will eventually die. And these 2 plants, they can fix nitrogen, but did not fix enough. So the plant still show nitrogen deficiency symptoms. Okay, you can tell this difference based on, oh, I'm sorry. Okay, if you genotype this plant, you know, this plant is plus plus, homozygous plus plus, and this plant is homozygous minus minus, and these 2 plants are heterozygous. So this tells us that heterozygous plant show intermediate phenotype when compared with the homozygous plant plus homozygous minus minus. You can also tell the difference based on nodule size, nodule color, as well as the microscopic structure of the nodules. Okay, so we use more than 10,000 plants and map the gene into a very small genomic region, 5 kb genomic region, which contains 2 genes that both encode a so-called nodule-specific cysteine-rich repeat, we call peptide, we call NCR peptide. Okay, and both genes are polymorphic between the fixed plus parent and the fixed minus parent. So we can consider these 2 genes are strong candidates for NFS1. Okay, so first I want to, before we talk about functional tests, I want to tell you what are the NCR peptides. Okay, it's they are 6 to 70 amino acids in length, which contain an N-terminal signal peptide, which help to secrete the peptide into the bacteria. And they also contain mature peptide, contains 4 or 6 conserved cysteine residues. Okay, this peptide bear similarity to defensin-like antimicrobial peptide in both plants and animals. And this represents a large gene family in legume plants such as medicago and alfalfa and pea, which subject their microbial partners to terminal differentiation. Okay, these genes are only expressed in nodules. Okay, so most significantly, these NCR peptides are required for bacteria differentiation and survival. Okay, in other words, these NCR peptides play a positive role in nitrogen fixation, because in order to do nitrogen fixation, bacteria need to be differentiated into the bacteroid, and these peptides are required for that process. Okay, so we test the function of the candidate gene in the near isogenic background of that residual heterozygous line. We cannot test in the original parent. Remember, we said, it's other gene also involved. So given that fixed plus to fixed minus to, shows 3-to-1 segregation. And our previous knowledge all point to that NCR play a positive role in nitrogen fixation. You could easily assume that if we transfer the plus allele into the fixed minus genetic background, we should be able to convert fixed minus to fixed plus, right? So we test the fixed plus alleles. So we transfer the first gene and the second gene from the allele of the fixed plus parent into the fixed minus genetic background. Okay, unfortunately, none of the two genes can convert fixed minus phenotype to fixed plus. Okay, and we also carry out CRISPR-Cas9 mediate gene knockout in the fixed plus genetic background. We knock out the first gene, we knock out the second gene. The mutant root didn't change the phenotype. Okay, the conclusion is that the fixed plus allele are not required for forming fixed plus nodules. So, what hell is going on? So go back to this picture, photos. When the heterozygous plant show intermediate phenotype, theoretically, there are 3 possible allelic relationships. Okay, plus allele could be partially dominant to the minus allele, and minus allele could be partially dominant to the plus allele. And alternatively, both alleles could be functional. Plus allele contribute to fixed plus phenotype, fixed minus allele contribute to fixed minus phenotype, and the heterozygotes show the blending effect of both alleles. Okay, a situation we call codominance. So based on the experiment I described at the beginning earlier, we can exclude these 2 possibilities, because if this are the cases, complementation test or CRISPR-Cas9 should give us phenotype, but we didn't observe such phenotypic change. So is that possible? Minus allele is a functional allele. So we go ahead to test this. So we want to knock off the fixed minus allele in the fixed minus genetic background. Okay, when we knock out the first gene, the fixed minus phenotype was converted to fixed plus phenotype. Well, knockout the second gene didn't show the phenotypic change. So this tells us fixed minus allele is actually a dominant functional allele. Okay, consistent with this, we transfer the fixed minus allele into the fixed plus genetic background. Okay, you see the wild type root is fixed plus, and the transgenic root are fixed minus. So this tells us the NS1 minus encode an NCR peptide that restricts rather than promotes nitrogen fixation. So this also tells us that NCR a double-edged sword for symbiosis development. Remember at the beginning, I talked about master-slave relationship. The plant uses NCR peptide to manipulate bacteria, to increase, to drive its differentiation, to increase the nitrogen fixation efficiency. But if the plant is pushed too much, some bacteria strain cannot tolerate. They die early, cannot do nitrogen fixation. Okay, so this peptide as this is alignment of the two peptide isoform. Okay, both peptides contain 6C. Okay, very conserved. That's why we call cysteine-rich peptide. There are 3 amino acid difference. Okay, so this 3 amino acid substitution is responsible for the functional difference between the two protein isoforms. Okay, and also this gene also is expressed only in nodules, and in the nodules is predominantly expressed in a zone we call interzone. This zone is where the bacteria start to differentiate. You can explain after they differentiate, they die quickly. So, and also we showed that these peptides are delivered into the bacteroid. So the red color is NS1 protein, the blue color is bacteria. So you can see the colocalization of the bacteria and the protein. So I don't have time to talk more. I just gave you a number of more findings from this project. So NS1 minus allele occurs at a very low frequency, about 8%, which suggests it's selected against in natural population. Okay, so plant in natural population, plant with the plus genotype can be fixed minus. This is because the presence of other genes. For example, we also clone a gene NS2, which share the same story in SS1. So if you have plus NS1, at one gene, if you have NS2 minus, it's still it's minus. Okay. So plants with minus genotype can be fixed plus, which means the negative role of NS1 can be suppressed by other peptides. So the take-home message is that the negative role of the individual NCR in symbiosis is dependent on host and bacteria genetic background. We can clone the gene because we homogenize the genetic background. Okay, so in nature, in natural population, whether a bacteria is fixed plus or fixed minus, is determined by a combination of a cocktail of NCR peptide. Okay, so with that, I want to acknowledge my student, collaborators who involved this project. Our funding came from NSF, USDA, and local funding agency. With that, I'll be happy to answer your questions. Thank you.
Yeah, so I have two questions. One is, do you think that the bacteria are activating a toll receptor that leads to the production of this peptide, the NCR peptide? Like,
Good question. I don't know. I should have learned the talk from Jack, so we have no idea.
And do you have any idea what the peptide is doing in a positive way to help the bacteria instead of hurt the bacteria?
We just know the bacteria, the peptide are required for bacterial differentiation, but what is the bacteria targets of the NCR peptide? I don't know. Michelle, you know that? No, it's tough to study because the function of the peptide is dependent on the genetic background. And you cannot single out that peptide in doing experiment in vitro to test the function, because you single out, you lost all the genetic background, because the function is dependent on the background.
So receptor-like kinases are evolving just like how our genes would be evolved, like CF9 locus, looks all the same. So that's a couple quick questions. One is, are there other receptor-like kinases in these loci that are resistance genes, or is this locus always?
It's a good question. So the NS2 locus, remember the large gene cluster, and this gene cluster is not legume specific. It conserved in legume as well as in non-legumes. Okay, in Arabidopsis is the syntenic region also contains this gene cluster. And many of that gene are involved in plant microbe interaction.
And so then these RLKs, are they acting through similar downstream pathways? Are they acting through MAP kinases and ORT transcription factors, that kind of thing, or is it different downstream processes?
Yeah, so that's a good question. We should, we assume that, for example, this surface receptor, recognize something to induce immune response, right? When we perform RNA-seq, we cannot find specific enhanced expression of defense-related genes.
PR genes are.
This is because, remember, the gene is expressed only in a limited number of foci. And if you do, the immune response only occurs in that small number of cells. If you use whole room, total RNA for RNA-seq analysis, the signal got diluted. You can't see the difference. So right now we propose to use a single cell RNA-seq to solve this problem. And also, we also apply candidate gene approach, as just mentioned. If the pathway involved in MTR, the molecular pattern triggered immunity. So we can also use candidate gene approach to knock out those genes to see if we can convert incompatible interaction into compatible.
So the locus at the end has the two NCRs. So the second NCR, do you have any speculation? Does that, so it doesn't play a role in this phenotype? Do you think it plays some other bacterial interaction phenotype role, or like it seems so odd that there'd be two in the same locus?
Yeah, we have no idea, but NS2 is not involved in the fixed minus phenotype for that specific bacteria strain. So whether it can have effect on other, I don't know.
Almost embarrassing basic question.
Mhm.
So when you were in the introduction explaining like the beginning of this relationship with the plant, the bacteria was just living in the soil beforehand. How does it all start? Because I didn't see the cycle closing up because it's like a very terminal differentiation for the bacteria. So how does the cycle close?
I understand your question. If we call it symbiosis, it should benefit the bacteria. If you kill the bacteria, it should play a negative role in the soil populations. Okay. So if you look at the metacargal nodules, which is indeterminate nodules, I mean, there are different parts of the nodule at a different stage. So in the nitrogen fixation zone, the bacteria got killed after nitrogen fixation. But in the infection thread or in other part of nodules, maybe also bacteria can multiply to increase their fitness in the soil.
How do they go from one plant to another? Do they have an intermediate phase in the soil, or can they survive outside of this?
I think so. Yeah, they
Yes,
can.
absolutely.
They have two different life forms.
Yes.
Yeah, saprophytic. They can also live in saprophytic. Yeah. And also, once you mentioned, we talk about NCR peptide. This peptide only present in cool season legume that drive the bacteria to terminal differentiation. In soybean, there's no such gene, no single such gene. Okay.
More questions? All right. So join me to thank Hongyan one more time.
Thank you.